What has happened? Okay, now I'm ready to go. Good morning, everybody. Hope y'all had a good weekend. I hope Packback treated you well. I saw there was a massive amount of posts. I've looked through a good chunk. Initially, I was like, wow, sweet. It looks like almost every single person has done stuff. But then I realized I think it's counting your questions and your responses. So I'm like, there's still a chunk that maybe didn't do some stuff, but that's okay. So we will in the very near future do a little bit of in-class polling. So basically the deal with that stuff, and I haven't actually done it yet, but I know I can. The deal with that is there will be a question projected with a QR code. You'll quickly snap that and it will take you right to it. And then it populates all of the questions, anonymous, so we can get like real-time poll questions about specific things. I think it sounds interesting. I would like to try it. Whether or not it's something that I use a whole lot will kind of just depend on how I think it functions for me, but it's supposed to be pretty straightforward. All right. So keep that in mind. And that if you are tuning in at a later time, it doesn't matter if you are not answering these poll questions. It's for people real-time Zoom or in class to pipe up with a little bit of feedback. So don't feel saddened. All right. Any questions? Any packback concerns? Okay. I left off Friday. We got pretty close to halfway through chapter 2. We should be able to wrap this up this morning. So thinking about all sorts of different potentials to examine microbes, employing different types of microscopy techniques. We talked a little bit about staining, and we're going to go through some more details with staining in a minute for actual real stuff in the lab. But this procedure is something that I think is pretty cool. We haven't thought about yet any immunology, but we will go through that unit of material and talk about antibodies, talk about cells that are producing antibodies, and ultimately an antibody is specific against some piece of substance that our body has recognized as being foreign and is like, crap, I ought to do something about this. And it starts pumping out antibodies to stimulate all sorts of different responses, mechanisms to truly destroy or to kill whatever that stuff is. All right. So antibodies against specific stuff opens up the idea for all sorts of different things that we could use them for. We can use them for detecting disease. We can use them in a diagnostic test to link up with something. So there's ultimately a little bit of biochemistry that happens in the background before you do this stuff. And they take an antibody. It depends on the situation. That gets labeled or tagged with some sort of a substance. In this instance, it's a substance that will fluoresce. There are other potentials where you might see some color changes or just the emission of light. Numerous different things could be tagged to these antibodies. Now this antibody does more than just bind. It tells you that it bound, right? That's important, all right? So here we've got a handful of different things. An antigen, our little spirally guy, could be syphilis. We talked about treponema a second ago, maybe, but this is just an antigen. All right. And then up above, a couple of different antibodies, a secondary, a primary, and then our, in this case, fluorescent tag, a fluorochrome. All right. Depends on the circumstances. All right. Direct and indirect depend on the type of antibody that's being utilized. In some circumstances, that first antibody against that specific thing, whatever it is, it might be able to be tagged. And then that would be a direct detection. This antibody links up to that specific antigen and it glows. All right. That's our first box up here. It's a little blurry, but these little green speckles in here are Neisseria gonorrhoeae. All right. A gram-negative cocci. All right. So imagine these are circles because they sort of look that way. All right. And the antibodies linked up to these cells and they glow. Now, maybe we gram stained and we saw these gram-negative cells, but we're not certain that that's who it is. Staining it with this fluorescently tagged antibody links up to it, and you know, because those antibodies will only link up to Neisseria. All right. So you could verify it that way, or simply just use it in the first place. But you likely would do some gram staining first. All right. This next one, couple of little parasites up here. These are schistosomes. These are not the substance that we would see the adult stage in us. We'll talk about that later. This is the stage, we call it Cercaria. This is the stage that would be infective to us. So they would swim around in the water, find our skin and penetrate in, and then they cause infection. All right. So these are what's getting us. All right. And in this case, this is an indirect. All right. So there's antibody, and this could be something that the patient has produced or whatever it might be. All right. But it's antibody against these schistosomes. The secondary antibody has nothing to do with the schistosomes. It has to do with the first antibody. So it's going to link up to, like I said, maybe it's something collected from us. Our serum sample, we've got antibody. If we have antibody against it, this antibody recognizes our antibody, links to it, and then it glows. All right. So it has nothing to do with the antigen. It has everything to do with that respective antibody, whoever it was created in. All right. And in a lot of diagnostic procedures, oftentimes these first antibodies, primary antibodies, are created in some sort of an animal, like a goat. They can produce a good amount of antibody. You can take blood from them, just like you were going to go to Biomat or wherever and donate some plasma. It's the same sort of an idea. They collect the blood, pull the plasma out, put the blood back in, and now we've got this substance that harbors sorts of different things, and we can purify it out. All right. And potentially label it with respective tags. So it depends on the circumstances. We will touch back on some of these mechanisms for diagnosis when we jump into immunology. So we'll see a little of this again, but I think it's really pretty impressive. Another technique is confocal microscopy. This is something that we think about kind of a potential maybe for some real-time visualization, situations where cells could still be alive. It depends on the situation. Oftentimes in micro labs, those cells get fixed to a slide, basically killed, as we get them cooked onto the slide and then stained up. This gives you the ability to see things potentially moving around. So here we're looking at some biofilm with some cyanobacteria, and we've had a little bit of staining to allow them to show up. I always tell this story in here because I think it's interesting. I believe, I'm almost positive, we have a confocal microscope in the biology building now. It was something we acquired a few years back, if I remember properly. These microscopes are very different, all right? And they rely on lasers to give us these images. It's not like rolling in and seeing a typical microscope with eyepieces that you look in. It's all kind of digital. You're looking through a monitor at what's actually happening in this machine. So my first experience with a confocal microscope was many, many years ago. And the microbiology club here took a field trip to a National Institute of Health lab. The government has two big research facilities, NIH and CDC. And there are a couple of satellite offices or facilities, one of which for NIH is in Montana, not too far of a drive.
Really, Sam?
All right. That's not too far. All right.
We could like you on the exam,
So
and then
we took this field trip and had the opportunity to see, spent the whole day up there.
Then I'll just go back.
The facility. This was just a couple of years before they opened their BSL level 4, so Ebola studying type of stuff. The lab that I feel terrified just to walk past, right? You're just like, holy cow. Not a job that I really would like. All right. Spacesuit sort of stuff. All right. We'll see.
And he does basically essentially.
All right. So as we get up there, we're going through, we're seeing all of these different labs, lots of different research, a lot. I mean, they do all sorts of things up there, but a lot of it initially was the idea of studying and understanding tick-borne illnesses, all sorts of different things. And we'll talk about Lyme disease a little bit later with some of that. So as we're moving through, we get to this one lab, and it's the confocal microscopy suite. We walk in, and it looks kind of just bare bones, all open, and just a little bit of stuff on this one wall, and we can see weird things along the two other walls. We walk in, they tell us what's up. We got a bunch of lasers going this way, and then the machine. Right?
And so I was like,
as we get in, this guy comes in his white lab coat with two syringes, and he looks mad scientist-like, right? With these big-ass syringes, right? And he asks us all, who's seen a macrophage eat some Staph aureus? Well, none of us, right?
You can see that.
You can see it now. YouTube has all sorts of good things, right?
What you eat during pregnancy can.
This machine has two little ports, and he injects both of these things into the two separate ports, and up on the monitor, like what we got going on right here. And I will paint this picture. It's not the same thing, but imagine these little pinkish guys in here were the staph, and the gray stuff right here was the macrophage. It starts off with a blob of pink stuff on one side and our macrophage on the other. Staph doesn't move. They just hang out. They're just sitting there minding their own business. They're not doing anything, right? But the macrophage, as soon as it gets in there, and it looks like it's forever away from them, but I bet you it's super close. I don't know. All right. All of a sudden, that macrophage smells the staph. They move by chemotaxis, so they can sense these things, smell it. There's something over here. And they move kind of like an amoeba. They project out their cytoplasm and pull themselves. And all of a sudden, this macrophage kicks it into gear, pushes itself, pulls itself with its pseudopod right up to that glob of staph cells. And all of a sudden, it engulfs, pulls it in, and you see this little cell, and it pops to the inside. It just phagocytized it. It just ate it. Like, holy cow! Awesome. All right. That's why I'm up here teaching microbiology. I'm just like, that is some
whoever has a mic on, can you turn
right?
on?
And we see it again, right? And the color just went from this guy's face. He's like, it took me 5 hours to prep these samples. That's all I got. I'm like, shoot, you should have pushed record, right? Oh, I'm getting some chat. Oh, somebody needs to mute that's watching on Zoom. Please. All right. Maybe I'm muted. We don't hear any of that. That's fortunate. Okay. I assume they hear me, or there'd be more chatting. They're like, what the hell? All right. So that was my experience with it. I will tell you one more tidbit, because before we went up there were some other students that had taken the tour a few years back, and they told us all, make sure when you get up there that you let them show you the moat. And so before we ever even get up there, I've got all of these pictures going through my mind that this facility is like some old school castle with a moat. And I'm like, what are we really getting ourselves into? What's in this moat? What are they trying to do? Right? As we got there and got into the facility, little did I know, I stepped right over the moat and didn't even know it. All right. The moat was this little, it's not like the gutter out at the street that's just a little dip, but it pretty much was. All right. It was concrete, a concrete channel. All right. Probably only about 3 or 4 inches deep. What the hell's going to happen with that is what I wondered after they actually showed us the moat. Back in the day, when the moat was potentially needed, not potentially, when it was needed, in a research lab, if you're studying infectious things and maybe ticks that are full of disease, you want to keep those ticks there. And you don't want them getting out. And this NIH lab is right in the middle of town, right? So you want to keep your ticks where your ticks are supposed to be. And ticks are all over the place. So you don't want any outside ticks getting into your facility. You got to keep those suckers out. So this little teeny moat, about a foot wide and a couple of inches deep, was full of oil. And any tick that's crawling around and hoping to crawl in or potentially crawl out can't, because it won't survive that little bit of oil. Fascinating. Now there's different things in the labs to ensure they stay in here or they don't come in one way or the other. The moat is empty. All right. Interesting. All right. So, and like I said, we'll talk more tick stuff a little bit later. But it was interesting. I thought this moat was going to be gigantic, right? Probably piranha, maybe some alligator, right? I don't know what would be in there, but something to deter people, right? It wasn't deterring people. It was keeping the ticks where they should be, right? Crazy, right? One guy had a tick in a little terrarium type of thing, just living there. She had been his little pet for a super long time and just hanging out in there. It had been 20 years since she had had a blood meal, and she's still in there, right? And I'm like, what? That's nuts. How long can a tick live? Pack back, right? I feel it. All right. And I don't know the answer to that, but it was impressive. It makes you think about all sorts of crazy things with some of these arthropods. All right, enough of that. So this is an image of an electron microscope. This is a transmission. There's 2 types of electron microscopy, scanning and transmission. I'll show you some images in just a second. This transmission microscope it's not the same, but it looks very similar to the electron microscope that we have in the biology building. Our microscope is pretty old, 1970s or maybe early 1980s, pretty old. All right. It was, as it was made, if you wanted to take an image, you took an image right at the microscope on some photography film, and then that film would go to a dark room and it would get developed. That takes a bit of time. And it also, many of us, probably almost everybody in here, just digital cameras, right? But the old school cameras, the 35 millimeter with the film in there, you would take a picture and not know if that picture really was very good or not. And lots of times some blurry crap, right? Like, well, that one didn't turn out very well. Now you just delete, right? Different, right? So that was the way it was for a super long time. All of that stuff, as we advance, they couldn't get the film they needed for that. It kind of got mothballed. And then a donor gave a good chunk of money to retrofit our microscope with some digital cameras and the ability to link it up to some computers to visualize on the monitors, just like we're seeing here. These are the oculars that they were using before. They still use this for the first little bit to see, and then it's basically flipped over to the monitors. And you can snap the picture digitally, done deal. All right. And ours, the top of it is kind of red orange. Bengals, right? I think that's just the way it was. All right. But I like it. I think it's kind of cool. All right. So the electron microscopes, it's a beam of electrons. There's a big source of energy up at the top. A tungsten filament gets super charged up. They use liquid nitrogen to cool this chamber because it is super high energy. So as we are focusing and energizing this filament, a beam of electrons gets shot down toward our sample that gets inserted, or up here, our sample that gets inserted into the device. And it is on just this little teeny mesh screen that gets inserted in there. It's pretty impressive to see, all right? As the energy flows through there, then down below where our lens is and we focus in on it, we've got the ability to visualize our specimen. To do the transmission, we coat whatever it is with something, a heavy metal. We are currently using some uranium acetate, which makes me think Simpsons. Trouble, right? But that's plutonium, right? That he takes home in his, right? It's different, but it's still uranium. Super kind of low concentration, but we technically don't work with that stuff. That goes to the chemistry department, to the radiation safety lab, and there are people there that do the staining for us. They are trained to work with that stuff, all right? Interesting. As the energy hits that, if it hits the material, it reflects that off, and it's that image that we're able to capture, all right? Whatever it might be, all right? I should have an insert of our bacteriophages right there. I dropped the ball there. That would be the perfect plug. I'm already talking about it. All right. So transmission and scanning. With the scanning, the idea is more like taking a specimen and shaving super thin sections of it and then being able to visualize insides of stuff. It depends on what you're truly trying to look at as far as which microscope you would be using. So here, our transmission, this, every time I see this, I'm like, that's a mitochondria, but it's not. This is biofilm, all right? So it's handfuls of microbes together forming kind of this community of cells. Next door with the scanning, these are Staph aureus, clusters of grapes. And this is like, I think of some food stuff with artificial coloring, right? And preservatives, right? That's what's going on here. These cells don't look purple. We don't stain them up like this. It's kind of more black and white sort of image that you get with it. But to make them pop, they go in there with a computer and artificially color them, all right? They color enhance. And Staphylococcus is a gram-positive, so it would look purple under the microscope with regular staining techniques. So they've colored it purple. I like that. And it looks like a cluster of grapes. And here I think that's what we've got going on. In the background is the mesh screen that the cells get adhered to and then stained up. And there's little, very little pores in there. And you can see the pores in the screen. And then this, if you were to see the screen that it goes on, it almost looks like a solid little piece of metal, but it's actually like window screen with little teeny pores in there that you can hardly see any of those because you truly need some microscopy to visualize it. Crazy. All right. So the biofilm, there are numerous bacteria that have potentials for some biofilm formation. When we think about disease organisms, this is a big deal because it gives that organism or organisms, if we're participating in some community sort of stuff, it gives them the ability to glue themselves to some sort of substrate, to take hold, to start replicating, and truly to start establishing a disease process. It can be bad news. The plaque on your teeth is some biofilm formation. All right. So it starts off with an attachment. Those that can form biofilms have a sugary coating, a capsule layer that gives them that stickiness to adhere to something. All right. So they attach, they start gluing themselves down. That's this irreversible attachment. They've concreted themselves to this foundation, and now we got something to build upon. And it's a sticky substance. Others can utilize that, and we can create a situation where there is a community of microbes working together, causing some cavities or whatever it might be. All right. So they start growing and replicating. That's our maturation phase. As it continues, this is where we're seeing that electron microscopy image, this big mass of mature biofilm, maybe some slimy stuff that you could recognize. Gross, right? As it matures, well, now there's the potential to maybe ship off some of our progeny, and the dispersion phase, part of that coating encasing them all there, kind of loosens up and it allows some of these bacteria to move away, to potentially go back to stage one and find a new suitable area to encase themselves, and another biofilm starts to form. All right. So at this point, you can see the little flagella up here. These are planktonic. They're actively moving and swimming around versus the attachment phase. And now sessile, we're not moving. All right. We're just growing. All right. Kind of cool. So here's an image. This is with some more fluorescence microscopy, the DAPI staining. These are some bacteria in a biofilm on stainless steel. That's a potential issue to me because you typically think of stainless steel as being lots of metal, being pretty antimicrobial. Well, how are you going to be able to grow in those circumstances, right? What is that truly providing? And in these instances, really, it's more just a substrate for attachment. They're not feeding on stuff. They're feeding on other things, potentially on this stainless steel. Maybe there's some biological material that wasn't cleaned off, right? And in turn, the bacteria are able to attach and start growing, right? That's the idea. You would hope if you're going in for surgery, that scalpel has been autoclaved and there aren't biofilms on that sucker, right? Or the tattoo needle. All right. You don't know. I feel like if you're rolling into a tattoo shop and you don't see an autoclave any place around, you maybe ought to look around. I assume you probably saw a little benchtop autoclave. You're good. All right. I assume, right? And if it's coming out of some dude's basement, he got to question that, right? Where's your autoclave? He's like, it's fine. I wiped it on my jeans, right? That's like some prison tattoo. All right. So quick table, little bit of comparison. I'm not going to spout off all of this stuff. The bright field microscopy, as we started this is the common thing that we're going to think about to visualize microbes. All right. With that said, in principles of microbiology, you really only have the opportunity to use the microscopes a few times. We're going to look at some stuff. We're going to do a bit of staining. You're going to get an unknown. You're going to stain that. There'll be a few times for staining. Probably some practical stuff where you need to do a little bit of staining. All right. But ultimately, just a few times. If you were to progress on into some other microbiology labs, it might be something that happens all of the time. All right. I teach a pathogenic microbiology lab, and basically that is something that they will be doing every lab period. They've got to stain something. They get something, they have to stain it to see what do we have going on, oftentimes a mixture of things. And then based off of what they see, how do we plate it? How are we going to grow it? How are we going to figure out what it is? Once you've got it to grow, you got to stain it again to see who is it, right? Which one is growing here or there, or are they both, right? Multiple things to initially see it, then to recognize it again multiple times. All right. So keep that in the back of your mind. If staining things and microscopy is something that you're truly interested in, there are some other potentials to really kind of dive deeper into that process. So the dark field, switching it up and taking the light away and the organisms showing up from glowing almost, if you will, as the way the light is hitting them. The phase contrast gives you the potential to see some more cellular details, surface details with those cells. And although we're looking at some bacteria here, it's oftentimes maybe a little more important if we were looking at eukaryotic cells than bacteria for phase contrast, because that gives us some greater potential for some structural differences. All right. The differential can show more kind of surface sort of stuff. We showed, again, eukaryotic cells and seeing more surface detail with them. The fluorescence, confocal, and 2-photon giving us potentials to zoom in more. We didn't talk about this, but this is kind of employing staining and electron microscopy, but not exactly the same. And then our last with the transmission and scanning with some Ebola, terrifying, and Campylobacter jejuni. This one is a bacterium that is very common in cattle and something that can be found in the milk and can cause some serious intestinal issues. There are many bacteria that we might be able to see in milk. All right. And you got to think for a minute about a cow out in the pasture or the corral, and the udders on that cow hang pretty close to the ground. There's a lot of manure all over the place. The potential for fecal contamination ought to be a given. All right. Today, when we think about milking cows, it's a little bit different than just rolling out there with your bucket and milking, right? They're a little bit cleaner in that process, but we still pasteurize, all right? You pasteurize, one of the main reasons is Campylobacter, all right? It's a bad one, all right? There are a few others, but Campylobacter was definitely one of the main things that we can thank good old Louis Pasteur for helping us to prevent if you're consuming milk in the first place. Which makes me wonder, why the hell would you? All right. Scanning and atomic force give us the potentials to zoom in even more. Here looking at some images to potentially see DNA, but this we're looking at gold all lined up, the respective atoms linking together, and more with some cellulose, nanocellulose. So I mentioned a second ago that when we're dealing with our bacteria, they're going to be stuck to those slides. You typically need to fix a specimen to get it to adhere, if you will. And there's a lot of different ways that we may do that. I kind of feel like we're missing one main thing. When we deal with stuff, we use the Bunsen burner. We use a flame to fix those slides. But the first two techniques that we see here could also be used. This is a slide warmer, and this is an incinerator. So it's like a Bunsen burner, but a little bit different. And if you were working with really hazardous material, microbes that could potentially aerosolize and cause, infectious disease that way, likely not going to use a Bunsen burner. You would use an incinerator, and your loop that you would be sterilizing would go inside of this chamber, and anything that aerosolizes stays in the chamber and gets killed. All right. If you put your loop in properly to the Bunsen burner, you also minimize the potential for aerosolization. But if it's truly a serious pathogen like that, these would be what you would think about. So there's heat coming out of this. All right. It's like a little toaster. All right. So in either circumstance, thinking microscopy, you're going to take a sample. It may be coming out of broth, or it could be growing on that Petri dish. The way you apply stuff is going to be slightly different. If it's a broth suspension of bacteria, the first thing I will tell you, if you're dished out tubes, you want to take your tube holding the glass, not the cap, and give the tube the littlest flick to mix it up. All right. That way, any bacteria that's potentially settled to the bottom, dependent upon when the bacteria was put into those tubes, it could kind of settle down. You don't want to just be dipping broth. You'd like to get organisms on your loop. Probably 3 or 4 loopfuls of broth. Apply those to the slide, kind of in a circle, move it around a little bit, spread it around, creating a smear. All right. If it's coming off of solid material like a Petri dish, a little scoop of the bacteria gets placed onto the slide. Your inoculating loop, you will put a drop of water on the loop, not a drop of water onto the slide, a drop onto the loop. Take that drop of water on your loop, touch it to where you put the bacteria, and mix it around. Now you've done the same as that broth stuff. That drop of water and your glob of bacteria is about the same as 4 loopfuls of just broth stuff. All right. Because there's a boatload in that little glob you put down. All right. So you spread it around, you make a smear, and then you let that smear air dry. All right. It's got to sit on the bench for a couple of minutes. And as the water evaporates, it starts to kind of look a little bit cloudy. All right. Then you heat fix it, either putting it on either of these 2 ways or passing it through the Bunsen burner. All right. There are clothespins that you can hook onto your slide and pass it through that way, or you can use your fingers. I prefer to use my fingers because I can tell if that slide is getting hot or too hot. All right. I don't want to burn my fingers. All right. But I would like to know that I have fixed it. So when I feel the heat on my fingertips, I'm good to go. Typically 3 passes, just like that. I don't hold it in there. I'm not roasting marshmallows. All right. I just pass it right through. If you're using the clothespin, same thing. Don't hold it in there. I saw somebody almost catch their clothespins on fire. They were smoking, right? If your clothespins smoke, you have fried those bacteria, all right? You want them to just glue on there, right? If you cook the crap out of them, you're going to blow those cells up and you're not going to see anything, all right? you just gently heat fix it. It causes them to stick, even though air drying fixes them a little bit. It causes them to glue themselves onto that slide better. Because once we start staining, you're going to add stuff, rinse it, add stuff, rinse it. All of those rinsings potentially wash away something that hasn't been fixed. All right. So that's the idea with that. Tissue samples can get prepped and preserved in a fixative like formalin. These things oftentimes would get sliced up super thin and then looked at through a microscope, dependent upon the situation, compound, or maybe even electron microscopy, just depending. All right. So few staining techniques. Anthrax, we saw images of anthrax before. This is a gram stain or part of it. And we can see the crystal violet that absorbs to these cells. So there's a lot of different ways that stains can act. And in this case, our crystal violet is a basic positive stain. The cell walls of bacteria, for the most part, are negatively charged. So positive stains, like a magnet, are attracted to the negative cell and they want to stick to them. All right. So, you know, and whether it's basic or acidic, whatever, but the positive, all right, sticks to it. Here we've got an acidic, but positive. All right. So it's sticking to it as well. All right. Different type of stain, pink versus purple. All right. And then lastly, with a different bacillus, this is Bacillus megaterium. This has a negative stain. So this is negative red. The background, the slide gets stained, and the negative stain and the negative cell wall of the bacteria, they repel, right? So the bacteria doesn't get any stain on it. It stays colorless where the background picks it up. India ink or nigrosine is another negative stain. It stains the slide and not the cells. Maybe a technique you will do in the lab. I think probably not. So this process you will do. And this is something that is really kind of gold standard when we think about microbiology. Doesn't matter if we're thinking teaching labs or a clinical situation, whatever, all right? To initially try to visualize cells, this is one of the first, maybe the first technique that's employed. There are other things like this previous image where we could just simply add one dye to our slide and see something, right? But if you've only added one, really all that's telling you is some details about that cell, the shape, the arrangement, right? A little bit of morphology idea, but it doesn't clue you into other details. That we call a simple stain, just the addition of one dye or stain, all right? And in these cases, all of these could be classified that way, even though I said gram stain, gram positive, right here, it just says crystal violet. So maybe that's all they applied. Even gram negatives, if all you put on them was crystal violet, they would look purple too, all right? But not if you do a gram stain, all right? So the gram staining procedure was created by a physician, Hans Christian Gram, developed the gram staining technique, hence gram. Multiple steps. You make your smear, you heat fix it, you've done all that I just described, and now you take your slide and you apply crystal violet. We call this a primary stain, and just like that previous image, everything that touches this crystal violet is going to turn purple, all right? The positive look positive, the negatives look positive, all right? So purplish blue color. And we've got different morphologies up here just to show us that, all right? The next step after you've added the crystal violet, and we're not showing details in here, but in between each of these steps is a rinsing with water, all right? Each of them need rinsing, all right? So crystal violet, rinse, add iodine, rinse, alcohol, rinse, safranin, rinse, then you view. All right. After you've added the crystal violet, you put iodine on there. Grams iodine is a substance that gets in with the crystal violet, and they interact and complex and form a big honking molecule that gets stuck in the cell wall of gram-positive cells because their cells are super thick, right? So it gets tied up in those thick layers of peptidoglycan, the cell wall material, and it sticks in there. So we call it a mordant because it interacts with the primary stain and it sticks it in there, all right? The gram negatives, they still look, and each of these is like, what if we looked at them under the microscope right now, all right? The gram negatives still look purple, all right? Because we haven't removed any of that dye, all right? The next step is the alcohol step. We use grams alcohol to decolorize. There's two different types of alcohol that get used in microbiology labs, grams alcohol and acid alcohol. Acid alcohol is same concentration of alcohol, but hydrochloric acid gets added to the mix, all right? It's used in acid-fast staining. It's used for staining procedures for like tuberculosis. You don't use acid alcohol in a gram stain or everything will now look gram negative, all right? It will pull the stain out of everybody, all right? Grams alcohol is what you use, all right? In these procedures, oftentimes lab manuals talk about this stuff and they say 5 to 10 seconds. Every other step is a minute of adding the iodine, adding the crystal violet. You let them sit on there for a minute, give or take a little bit, all right? You don't have to set a timer, all right? The alcohol step is critical, all right? You don't want to add too much alcohol. Can you ever have too much? Yes. You don't want to add too much alcohol, and you also don't want to have not enough, all right? It's important to watch your slide, not the clock. Don't wait for 10 seconds because it said 10 seconds, all right? You take your slide, at an angle, you will have your alcohol squirter, and right above where your smear was, you're going to squirt the alcohol on there. I'm looking at the bottom of my slide as I'm squirting alcohol up above the smear. Initially at the bottom, I see purple color coming off, then it gets lighter, some blue, then very light blue, and then no more. It's just clear. As soon as I see the clear, it might be 3 seconds, it might be 13 seconds, just depends on how thick that smear truly was, all right? I squirt it on there till I stop seeing color, and I immediately grab my water squirt bottle and I squirt right up above where I was adding the alcohol, and I wash the alcohol off with my water. Don't trust the time, trust your eyes, all right? That will let you know, all right? If you go too much, you can make some of those gram positives look sort of negative. And if you don't go enough, some of the negatives are going to end up looking positive. Either over or under decolorized can be a problem. Looking at your slide will key you in. At this point, if we slapped this on the microscope, the gram positives are still positive. The alcohol doesn't do anything to them. The iodine makes it all stick in there. But gram negatives only have a thin layer of peptidoglycan and then an outer membrane made up of lipopolysaccharide. Alcohol hits that outer membrane and it dissolves it, makes it kind of wither away. And now it grabs that little bit of stain and pulls it right out of there. Only one layer thick, nothing to stick into or tether itself, all right? So now the negatives look colorless, ghost cells, and the positives still purple. We counterstain with safranin. It's a pinkish dye. Pink and purple don't do anything, just purple, all right? But pink and colorless now makes these guys able to be differentiated. So it's a differential stain, telling us the differences between gram positives, thick cell walls, and gram negatives, thin cell walls. Done deal, all right? Now you know more. You know morphology, arrangement, and cell wall characteristics. Why do a simple stain? When just a couple extra staining steps tells you a lot about who you're looking at. It doesn't, I can't look at this and immediately say, oh, that's got to be E. coli because it's pink. You don't know that, but you know it's a gram negative, right? Or Streptococcus pyogenes, gram positive and kind of chained up like this. I don't know that for sure, but my gut says that's probably who this is. All right, I will quit with that. We will pick up with the littlest bit of this and chapter 3 on Wednesday. Farewell, good day.